But now the next speaker is Rajan Sankaranarayanan. And Shankar will talk a little bit more about evolution. Shankar has been at Center for Cellular and Molecular Biology in Hyderabad for about 22 years. And his background, master's is in physics. After that, he joined Indian Institute of Science for his PhD with Professor Vijayan, finished his PhD, then went to do postdoc with Dino Moras, where he picked up the amino acid RNA synthetases. Here he was working on lectins, the very favorite molecule of Vijayan lab at that time. So he's got a lot of expertise there, as well as in amino acid RNA synthetases. He's doing really great work, and his work really has been recognized by a lot of awards. I can't tell you all of them because he's pretty much got every one of them. You know, Shanti Sarup Bhatnagar Prize, National Bioscience, J.M. Ramachandran, list goes on, J.C. Bose. He's also a fellow of all the 3 academies in India and doing really great work. He's one person that I have really admired in a way that, you know, from structure biology, he's gotten into hardcore biology and evolution. And you will see that example. You know, when he came back, he was working with Chirality RNA synthetase. In fact, Tanweer, was he your first student?
No.
Yeah, but he worked on Chirality RNA synthetase where he picked up this DTD, and you'll see a lot of that today. And give a lot of introduction.
Great, yeah. So, very good morning to all of you, and thank you, Umesh, for that kind introduction. Very rarely such thing comes from Umesh. So. Okay, so glad to be here. And actually, it's very nice that I am following the talk of Jack, considering the fact that he has taken the Darwinian evolution, as he said, out of the ground. So what we are going to see is just after what happens, correct, when it took off from the ground. So since I have 2 talks, I will be with you for another 1.5 hours or so. So get ready for that. Can you hear me at the back? Fine? Okay. So I have just divided my talk into 2 themes. And these 2 themes also kind of coincide with how our lab over the last 23 years has gone on at CCMB, as Umesh mentioned. So the first 10 years of our lab, we were more looking at it. We didn't think of starting a problem on evolution or anything. Our lab was a crystallography, structural biology lab to begin with. And we were interested in certain molecules as to how they do the so-called proofreading, which I will cover shortly. And wanted to understand the mechanisms of proofreading because they were just coming out around that time. So what we realized is that these molecules or these mechanisms that evolved had certain link to what I call as prebiotic, just at the late stage of prebiotic evolution to the biotic evolution, which covers the last universal common ancestors to single cell organisms. And the time frame that we are talking about is 4.2 billion years when life possibly started as a LUCA to 2 billion years pre-eukaryotic kind of scenario. So that will be the theme of my first talk, followed by the second talk where I'm going to talk about how during the process of eukaryogenesis, organisms like bacteria and archaea that have evolved independently over 2 billion years, they mixed. And that created serious conflicts with certain processes. And I'm going to talk about one of those processes because the molecules, even though they started off with a common origin, but they had cross purposes the way they have evolved over 2 billion years. And when they came together, what kind of conflicts that happened, which had implications for the emergence of eukaryotes. Without those optimization of the dynamics between the protein and RNA, such kind of evolution would not have been possible. So I will touch upon a couple of examples. So the second talk would cover 2 billion years till date. In fact, I should thank Umesh for asking me to present in a meeting like this, because this allows you to reflect a little differently the way you keep doing things, to put it in a slightly larger perspective. Thank you, Umesh, for that. So the first talk would cover these kind of concepts, because as Umesh again told us, we should start with some textbook material, because it involves people who are possibly from different area who may not know the background material. So I'm going to start off with certain textbook concepts of proofreading, how the idea of proofreading in biology emerged to begin with, followed by what we call as chiral proofreading. As all of you know, I will cover it also a little later, that proteins are made of basically L amino acids, with the exception of glycine, which is achiral. So we all assume in general, as biologists to begin with, or from outside people who come in like me when I came, we assume that biology is full of only L amino acids, because the proteins are made with only L amino acids. However, over the last 10 to 15 years, I should say, that the analytical techniques have improved so much, and people have found these D amino acids, the mirror image counterpart of L amino acids, across the board. And some of them are in as high concentrations as that of the L amino acid counterparts, and they continue to create a problem even today in terms of confusing the translational machinery or protein making machinery. So these errors have to be corrected, and which are happening as we speak inside our body or in any other biological system that is known. And therefore, we coined this term or introduced this concept called chiral proofreading, which I will be focusing on the first talk. And basically, since it is going to be more on the mechanism, particularly this talk, so I will just show the role of RNA in catalysis and how the early protein RNA hybrid catalysis could have possibly originated based on the example that we have. So here we are talking about real biological systems, and these are the error rates associated with, I'm sure most of you must have read. And these are, of course, error rates which are so high, I mean, so low rather, compared to the kind of error rates which Jack showed, because they were the very early systems which were just trying to begin the processes, whereas here we are talking about processes that are reasonably set in, and obviously, as you can see, the DNA replication has the lowest error rate that one can imagine, and for obvious reasons, because they perpetuate the genetic information, followed by many of the processes in the transfer of information during the central dogma. You do have errors which are of 1 in 10 to the power 4 or 5, which is reasonable to sustain the system. The process that I will be mostly talking about is that of this area, wherein the amino acid is coupled to a tRNA by systems called tRNA synthetases, and they make an error rate of 1 in 10 to the power 4 or at max of 10 to the power 5. So how these error rates are maintained, and what is the implication of these error rates? And as you can see on the right, that there is a need to preserve the genetic information so that errors have to be low, but you can't have without errors a system that perpetuates, because then you can't evolve. So it's a nice trade-off between how much of error that one can possibly allow. And what is seen nowadays, I'm not going into that area, what is seen nowadays is that how errors are getting modulated upon stress. So nature has a way of regulating the errors in such a way that these errors are not fixed numbers. So system starts allowing more errors in certain cases where there are needs. And when everything is, the situation is happy, then the errors are at a much lower rate. So that is how systems behave. Okay, so I look at fidelity of information transfer. I'm taking this little chronologically, and the first person to touch upon the concept of errors in biological systems is none other than Linus Pauling. He didn't talk about protein synthesis, he didn't talk about whichever biological molecule. He said that in biological systems, if you have to recognize substrates which are going to be. Are you able to hear me at the back? I think they are saying, okay, you guys have a problem. I can come. You just raise the hand if in case if you are not able to hear, or shout, okay? Oh my God. Okay. So you didn't miss much.
Now it is better. Now it is definitely better.
Okay. So I thanked Umesh profusely to begin with, yeah. So, okay, so the idea of error correction or the biological systems have to put up with these kind of problems came from Linus Pauling, because he just looked at from a pure chemical, recognition point of view. And what he said was that if there are molecules which are going to be very similar, selecting them is going to be a problem, particularly the kind of accuracies that are required, possibly during biological systems, transfer of information. So these systems have to have some way of improving their, error correction processes. So this is what he said in a very paper which is written in German. Then you had the concept of chemical proofreading that came. I'll go into that, which is, of course, by giants of science like Paul Berg, Alan First, and then Paul Schimmel later on. The kinetic proofreading as to how these processes can actually happen. The first kind of an example or a methodology by which this can happen was given by Hopfield way back in 1974. And that kinetic proofreading, kind of applies to both what happens at the ribosome, which I'm not going to touch because that itself, ribosome, what kind of proofreading happens is a larger issue. And what I will be talking about is more on this side, but the kinetic proofreading is a process by which such things happen. Now, this is very simple for all of you to understand. Here we are talking about only L amino acids. If you look at the structures of many of the amino acids, they are quite different, as you can see. For example, you take phenylalanine and glutamic acid. Therefore, if you have an enzyme which is supposed to pick glutamic acid, it's very unlikely to make a mistake with phenylalanine, or even if it makes a mistake, that's going to be way below the errors that are required for accurate transfer of information, as we just saw the numbers. However, there are amino acids like, say, for example, isoleucine and valine, and serine and threonine and valine for that matter. And of course, nowadays we know even for prolyl tRNA synthetase can mischarge alanine on top of it because alanine concentrations are much higher in the cell and so on and so forth. So there are issues of recognition by an enzymatic pocket which will not be able to discriminate these small substrate with an accuracy that is required for transmitting the genetic information. So that is therefore, this problem has to be taken cognizance of, and how biological systems solve the problem was the question. And as I mentioned, Linus Pauling was the first person to touch upon the issue of that these molecules are going to, biological entities are going to have problems in recognition. And if you just look at it from an enzyme active site point of view, and if you look at how these molecules can be separated in water and with respect to an enzyme, what people have shown is chemically the error rates can be of the order of 1 in 5. However, the observed error rates using cell-free synthesis system, what people have figured out way back is that these errors are much, much higher. Much lower. So the numbers are high. That means the errors are very low. 1 in, say, 3,500 to 40,000, which is way lower than what you would expect from a chemically discriminating system. So how these synthetases achieve such a high specificity was the question. And the first person to show this phenomenon was none other than Paul Berg. And what he did in a very beautiful experiment, which I will summarize here, I will not go into the details. If you take isoleucyl tRNA synthetase and put isoleucine and ATP and the tRNA, you will of course have ATP consumption very robustly because you are going to make, it's a two-step reaction, so you make isoleucyl adenylate as a first step of the process, followed by transferring the isoleucine to the tRNA isoleucine. That's a two-step reaction. However, valine can in principle be activated by isoleucyl tRNA synthase, and it can robustly produce as much isoleucyl, valyl AMP as that of isoleucyl adenylate, which was quite surprising. However, the transfer of valyl adenylate or the valyl moiety from valyl adenylate to the tRNA isoleucine was much lower. So therefore, the first step of the reaction is quite robust as that of isoleucine, which is a cognate substrate. The second step becomes very, very slow. So how does that happen was the question, but he first showed the phenomenon of a pre-transfer editing. What I mean by pre-transfer is tRNA is the molecule where the amino acid has to be transferred. So what is happening here is a transfer step which is before the transfer to the tRNA, which means you are transferring the amino acid to the ATP to make amino acid adenylate. So this is what was shown. And this was the birth of what is the concept of editing or proofreading in any biological system for that matter. And of course, Hopfield came up with a beautiful explanation as to how this can happen. So the idea of Hopfield is that the enzymes try to break these one final reaction into multiple steps. And you can have, I'm not going to go through all these, you can have an initial complex formation, and then you get into a product which is an intermediate to the product. You don't not go to the final product. And that would be an irreversible step in the reaction. So which means if you have made the adenylate, you can't reverse it back. So that intermediate now becomes a block wherein you can actually do certain cross-checking. And therefore, because the intermediate is staying a little bit more in the reaction center, you have a way of correcting those errors. And that's why it is called kinetic proofreading, which you do in 2 steps. And as I said, for isoleucine, the ATP consumption, isoleucine tRNA synthase, the ATP consumption for valine can be quite high, but overall charging is much, much lower, as seen earlier by Paul Berg. And then came a molecular level explanation for the biochemistry. And this came from none other than Alan First. And that was a very beautiful model wherein he invoked that maybe these enzymes have 2 active sites acting independently. One, he called it as a coercive, wherein substrates that are very different can actually be thrown off, but substrates that are very similar can go through this process and go to the second sieve, where the second sieve is designed. It's a very interesting concept. The second sieve is designed for a smaller substrate, and therefore a larger substrate cannot bind. So in this case, if you take, there is an echo here. You guys are okay? Okay. So if you take here, this step is made for anything bigger than isoleucine to be rejected. Okay, so all the smaller ones would go through. Here, the major substrate that would go through here is valine because of the structural similarity. So the second active site is designed for only valine. Therefore, now isoleucine would be bigger substrate, so it's easier to reject that. And therefore, it will not go through. So you are actually talking about a smaller kind of a editing pocket, wherein the cognate substrate will not sit, and therefore it will be rejected. I will come back to it as to why I am emphasizing on the cognate substrate rejection from the active site. You will know a little later. But this is the model that was proposed by none other than Alan First. And the biochemical proof of such an editing happens in biological system based on Alan First's model. Biochemically was shown by Paul Schimmel. Paul Schimmel told that in isoleucine tRNA synthetase, there is a site which is inserted into the canonical synthetase fold called Rossmann fold for class 1 tRNA synthetases. In that fold, there is an activity. That activity can clear valine attached to tRNA isoleucine. So he actually showed biochemically that which region of the protein has this additional activity biochemically. Then came the, the structure, which was solved by Shigeyuki Yokoyama from Japan, and his student Nureki now is a very prolific researcher in multiple fields. They showed that the editing site is positioned in such a way that if a CCA were to flip, maybe I have a movie, but unfortunately the movie didn't work, which we tried quite a bit here. So if you think of this as the catalytic domain, which produces both isoleucine as well as valyl tRNA isoleucine, where the CCA end is the one which captures the amino acid. After capturing either isoleucine or valine, this movie, if it were to work, the CCA end will flip to this blue colored region, which is the editing site. So the editing site would bind specifically valine, and therefore they solved the complex with valyl adenylate and showed that valine actually can bind. And in that pocket, isoleucine cannot bind because it's a bigger substrate, it will have clash. So the CCA flip was invoked to show that a mode of post-transfer checking after the amino acid is transferred, it just checks for the correct amino acid. This you can imagine a process which was somewhat similar to the polymerase proofreading. So the CCAN flips to the editing site where this activity is checked, and if it is the correct substrate, it will be rejected. If it is the wrong substrate, it can bind, and therefore it can be hydrolyzed. So which kind of provided a structural basis for Alan Firth's double sieve model. All of you are with me so far? You can ask me questions anytime. Yeah. Okay? Feel free to raise your hand. Microsoft is not responding. That's what it says here. Anyway, so this kind of structural work was happening around, and many structures were solved. So this was, I'm talking about around late 90s when the first structural proof came, and many people have solved for other synthetases, which we also did at some point. I'm not going into those details, and kind of established this kind of a double sieve model wherever there is a proofreading that is invoked. Can we? Yeah. Okay, great. So the mechanistic studies were going on parallelly. This work from the group of Paul Schimmel and Susan Ackerman came several years back, almost 20 years now, which was kind of path-breaking in the sense that generally you consider these are housekeeping enzymes. So if you have a mutation, it's going to be deleterious, they will be selected out. But here in a population of mice, they identified this so-called sticky cell mice mutant, wherein, very interestingly, the mutation was in alanine tRNA synthetase. The alanine tRNA synthetase would mischarge serine and glycine. I will come to it a little later. And here is an editing site which only partially compromises the editing activity. The mutation does not kill the editing activity, it only partially compromises the editing activity, and that was sufficient to create protein folding problem and eventually leading to unfolded protein response and cell death, leading to Purkinje cell loss, which are terminally differentiated cell, and ataxia. So this was the first proof that mutations in proofreading domains, even if they don't kill the activity, if they compromise the activity at certain cellular or tissue levels, you can have issues. So this was the first demonstration of the concept of the importance of proofreading in biological systems. So this is around late 90s, 2000, when I was looking for what kind of problems that I should be handling in my laboratory. So this was the time of expansion of genomic informations, correct? And a lot of sequence analysis were done, and the genome sequences is coming in. People were looking at across the board what kind of sequence conservation is there, what kind of variations were there, and particularly in the context of translation of the genetic code. And as I have listed here some of them which are very interesting. Particularly, there are certain organisms which do not have asparagine or glutaminyl tRNA synthetase. Okay, and people who not working in translation may not know this, including in mycobacteria. You don't have them, but of course those organisms do make asparagine and glutamine in their proteins. So how do they do? So people invoked something called a transamidase. So here the aspartyl tRNA synthetase is little non-discriminatory. It will put both aspartic acid as well as asparagine on top of tRNA as well as on asparagine. And there will be a transamidase which would specifically go and convert the asp on top of tRNA asparagine to asparagine. And therefore you can generate those kind of amino acids for protein synthesis. So that is how that process goes. And there are, for example, in first time, now of course we take it for granted that things in the genome can be spliced together. In fact, certain tRNAs were missing, and people found, say for example, this was the example where it was in nanoarchaeomucutans, as an archaea, where they found even a small tRNA gene was split into two different places, and it was spliced to produce that tRNA. Now we take it for granted, but at this time, it was quite kind of revealing as to how these things happen. So these are some of the variations that came in. And one variation from a structural and mechanistic perspective that we were excited because we were involved in solving the structure of an enzyme called threonyl tRNA synthetase, because it's a auto-translational repressor. I will not get into that. So we wanted to understand the mechanism of that. During the process, we figured out that threonyl tRNA synthetase also requires editing because it mischarges serine on top of tRNA threonine. Okay, and it has an editing domain. But interestingly, the editing domain is not conserved through evolution, as you can see. This is the editing module, which comprises of, say, 150, 200 residues. I was talking about the editing scenario in the case of isoleucyl tRNA synthetase, or say valine tRNA synthetase. They have an editing domain called CP1, but that CP1 domain is conserved across evolution, which means you go to bacteria or archaea or go to any eukaryote, you will find the same CP1, of course, with the genetic drift that you would expect them to have in sequence. But they all had the same fold and performing, one can imagine, function very similar. However, in this case, the archaeal editing domains were very different in terms of the sequence. Structure was not available, but for the bacteria, the structure was available. And when we compared, this was so different. So why a highly conserved process in evolution, like translation of the genetic code, should have this variation? And what is the implication of that variation is the question that we began to ask in the early 2000s when I started. And to cut a long story short, we solved the structure of the enzyme, because that's what we knew at that time what to do. So we solved the structure. And what we found was this protein fold. Right? We solved the structure and figured out the fold. Had significant structural similarity, not sequence similarity, then otherwise we would have figured out. After solving the structure, we figured out that it has a very high structural similarity in terms of fold to what is called D-amino acyl tRNA deacylase. Till that time, we had no clue of D-amino acids in biology. We went around and asked people also. They were equally puzzled, including in the biology guys. So this was really, really striking. And then when we went and looked back at the literature, DTD, the D-amino acyl tRNA deacylase, I will be telling in short DTD. That DTD enzyme is a freestanding enzyme. It's not attached to any tRNA synthetase. Was identified by none other than Paul Berg. I will come back to Paul Berg a little later in my talk or in the next talk. So it was originally identified way back in the 60s by Paul Berg. And it removes specifically D-amino acids charged on tRNAs. And therefore, you can imagine it to kind of ensure homochirality during the evolution, which I will again come back. And this particular domain has high structural similarity despite lack of any significant sequence similarity. Okay, so the fact that you have a D-amino acid removing fold attached to a tRNA synthetase in an archaea in a very covalent manner, correct? That whatever the enzyme that we figured out was a cis editing domain. So it allowed us to propose a kind of a model for perpetuation of homochirality. See, if you think of the early proteins as what the early replication with Jack was talking earlier, if you think of the early protein making, you can make once in a while mistakes, for example, isoleucine with valine or even with phenylalanine and so on and so forth. But that doesn't create much of a problem. But if you put a wrong D-amino acid, say instead of L-isoleucine, if you put a D-isoleucine or another D, that will have serious consequences, as we'll see, for structure and function. So the Ds are going to be much more dangerous in a growing polypeptide chain. And therefore, we proposed a model wherein the early synthetase systems, correct, the pre-Luca kind of systems would have made these kind of protein-based hybrids, which would move away D-amino acids from getting activated on top of the RNA. And that was the model for perpetuation, which we proposed long back. So that brings me to this topic of what we kind of termed chiral proofreading over the next 10 years to show the process of how active systems are in place in biological systems across the board, which takes care of chirality even today. As I said, we all know that the L-amino acids are the ones which are taken for protein synthesis, despite the presence of significant D-amino acids in biological systems. And of course, if I now tell these processes, you will be able to connect as biologists as to where these D-amino acids are invoked. Cell wall synthesis, invoke D-amino acids, chemotaxis in plants, and plants have multiple, kind of D amino acids which are responsible for different functions. I will come back to plants little later during the second talk of mine. And D amino acids are also well-known neurotransmitters. And some of their D amino acid concentrations are as high or comparable to the L amino acid counterparts. Therefore, they are expected to have a problem if you don't have chiral selection during the translation process. So the protein synthesis ensures, despite the presence, it is not an origin of chirality problem. It is a perpetuation of chirality in today's biological world. How protein synthesis proceeds is the issue. This is a cover of Science that came again 30 years or so back, wherein you can't biologically synthesize a D amino acid. Okay, D amino acid protein. So they chemically synthesized, Kent and co-workers, chemically synthesized what is called a protease molecule, which is in this case a HIV protease. So instead of using L amino acids, they did a chemical synthesis of a D amino acid peptide, which is a D amino acid protease in this case. And what they found was that it can fold, and then also showed structurally it had features which are very similar to that of L amino acid, except that the D amino acid protein would be a mirror of the L amino acid protein. Now the function of the protease also had become a mirror image, but now this D amino acid protein would not be acting on L protease, L peptides, it will act on only D peptides. So the protein as well as its function have become a mirror image of the L amino acid counterpart. And so, as I said, you can make D amino acid proteins, no problem. Why nature picked L amino acid instead of D amino acid is a larger question. That's a question of origin of homochirality. And people say that all the nucleic acids use riboses, which are of D configuration. And therefore, they have certain role in picking up L amino acids. Then that becomes a chicken and egg problem, right? So if why they prefer D, why they can't be L? So, but the current world of RNA world hypothesis, what we think is that if the RNA had a D sugar to begin with, they can actually push towards an L amino acid protein, correct? That's what is the current understanding. So you can make full protein with D, but however, you cannot make heterochiral polypeptides. In a growing D polypeptide chain, I can't put L amino acids because it violates Ramachandran's map consideration, because the Ramachandran map would be just an inversion of the L amino acid Ramachandran map. So it can actually break a helix and you can't form correct structures. So misfolding would occur and D amino acids should be excluded from the translational apparatus. So this is a review that I wrote. I'll show you at the end. If I forget, I have a list of references for people if they want for further reading. Just remind me, I have that list at the end. So this is one of the review that we wrote, that the chiral checkpoints are not at the level of only amino acid activation. The chiral checkpoints are now known across the board, okay? So the first and foremost, of course, the process is started by amino acid RNA synthetases, but even today, the current day synthetases make mistakes of this, which are which are higher than what is expected for translation. And if the D amino acids are activated by this, elongation factor can also carry the D amino acids to the ribosome, but their efficiency, chiral selectivity efficiency is 1 in 25. And this is followed by the ribosome, which also can incorporate D, but at a much, much lower rate. Okay, so now people know at a peptidyl transferase center as to where these chiral discriminatory features are. And therefore, each step of the process is porous for D amino acids. And therefore, now there are lots of synthetic biology efforts across the board in terms of generating systems which can actually make in a cell-free system or even within the cell D amino acid proteins, because that will have a lot of functional usage in the synthetic biology approach. And I know people, some of them are also worried about mirror life forms that are going to be generated shortly. So that can have issues. Of course, they have a lot of ethical issues. However, there is a mirror molecular biology versus mirror life form. So what is that we should go to and how far one should go are topics of current discussion. But the bottom line is here is that there are chiral checkpoints across the board, and these chiral checkpoints ensure, even though they are not highly selective or very highly selective, they are porous, and therefore they leave significant amount of D-amino acyl tRNAs at the end in the cell, which are dead end products. I hope I'm stuck here again. These are slides which are supposed to normally work. There is no movie or anything. Okay, so it's almost like if you have plenty of D-amino acyl tRNAs in the system, the protein translation stalls, correct? So maybe you wait for some time so that the actual hero comes into play who can clear this D-amino acyl tRNA so that translation can proceed. Yeah, please go ahead. Yeah. Sure. So, it's a very good point. I hope people could hear the question. Do all the synthetases have proofreading domains? The simple answer is no. Because as I said, there are amino acids whose chemistry is very different than the rest of the amino acids. They do not require. Say, for example, if you take original tRNA synthetase, they very rarely require any editing because they are very specific. The interactions are very tight. Aspartyl tRNA synthetase and so on and so forth. The molecules that I mentioned, the amino acids that I mentioned, like isoleucine, valine, threonine, proline, so there are almost 10 synthetases for which you have an editing domains requirement. And not all of them possess the editing domain in a cis manner, in the same sequence. Some of them have what are called trans editors. So you have a freestanding molecule, which is an editing center, but that would do after the charging event happens and after the tRNA leaves the enzyme. So they are called trans editors. So you have these two separations. Is that what you asked? Okay. Yeah, please. Come again?
Those editing domains.
Biologically,
cannot be made.
Sure.
Can make both. Several proteins can be made.
No, it's not at a protein level. So now you can, in principle, make in the cell certain tRNAs to carry more D amino acids. Okay, that's a possibility. But now they have to cross the other checkpoints. So the first checkpoint is what we are talking about here. So if you cross the checkpoint, no, sorry. Okay, I think why don't you sit here if you don't mind. So yeah, so coming back to your question, you will have more of D-amino acyl tRNAs in the pool, but then they also have to cross the checkpoint of elongation factors because they would continue to reject more of D-amino acid ones. Okay, so then if it goes to the ribosome is not going to put the second D. The first D can be possibly put, but putting the second D is much more lower. So with all the tweaking, what people have done today is to make, if I'm not wrong, 5 to 6 residue D-amino acid peptide using a ribosomal system. Because the efficiency goes lower and lower and lower. As you can imagine, the peptidyl transferase center, where the D has to come in, it will not be in the correct orientation, therefore it will be rejected. So there are multiple checkpoints which would disallow D amino acids to enter the ribosome for protein making. Okay, I hope I'm clear. So now, with all the errors that are made, can we hold on till the end if you don't mind? Because otherwise Umesh will say stop. We can also discuss during lunchtime, but if you have a pressing question, please. Okay, so these D-amino acyl tRNAs are hanging around in the cell and somebody has to come and clear. And that's the job of what are these proteins called DTD, which I said was originally identified by Paul Burke. But now what we know is that the DTDs have their own evolutionary history. So when we looked at the presence of DTD, what I'm going to be talking about in the first part of my talk mostly would be about DTD1, which is a sequence and a fold which is very unique and present majorly in bacteria and present across eukaryotes. Archaea has another one, I will come back to it later, and there are variants, and then how the eukaryote, the whole thing gets split, which I will come to in the second part of my talk, in the second talk rather. So the role of DTD is a simple thing. It has to go and look for the amino acids in the cell and remove it from the tRNA, the amino acids attached to tRNA. Now it looks very simple, correct? From the way to say. But imagine in the cell you have abundance, real excess of L-amino acid tRNAs of different types. And now this guy has to be so selective that it should not deplete the L-amino acid pool, which is present because they have already consumed ATP, and that will be a deleterious thing. So what it has to do is to specifically look for D. Do we have 19 D amino acid deacylases? Because there are 19 D amino acids have potential for creeping into this system. No, there's only one D amino acid tRNA deacylase which can work for all the 19 D amino acids. And therefore, it has to be highly selective for the configuration of the amino acid without looking at whether the side chain chemistry is arginine or aspartic acid. Correct? It can be as different as that, but I am going to look for only the configuration which is around the C alpha atom of the carbon of the amino acid and go and look for the problem. And that is the challenge which we thought we should look at from a mechanistic point of view, because that was not very clear or obvious. So a single DTD suggesting a chirality based selection, that's why we call this methodology as or a concept as a chiral proofreading methodology. And so we solve this structure of this enzyme in complex with the tRNA substrate, which is a D amino acid one, nearly 10 years back. And the structure and mechanism turned out to be very beautiful. I'll explain to you in a minute. Yeah, let this rotate. So we are now focusing on the active site. And the first and foremost that you can notice, this is of course solved with the D-tyrosine. So the amino acid side chain is kept just outside the binding pocket, correct? Because it didn't care whether it is incoming amino acid is tyrosine or arginine or whatever. It has to not worry about it. And there is one, it's a dimeric entity. You can see the role of the green coming into the yellow subunit, while the yellow subunit binds most of the tRNA and the final end of the substrate. The chiral chemistry is looked at by an element which is coming from the other monomer, which is shown here. I'm not going into the details, otherwise we keep going. So there is a gly-cis-pro. Notice the cis-CAS, where the general peptide that you see is a trans peptide, right? But when gly and pro are involved, you can have cis peptide formation. For the structurally oriented people here, what we mean by cis here is the omega angle. Other than phi and psi, there is something called omega. That's a peptide bond, the dihedral subtended by the peptide bond. And that can be 0 or 180. When it is 180, we call that as a trans peptide bond. That's what we see in majority of the polypeptide. But occasionally you can have cis peptide bond formation. So this is a gly-cis-pro entity, which is conserved across evolution, starting from bacteria to all the way to humans. I'm not going into that. And that is inserted into the active site from one monomer to the other monomer, wherein it captures exactly the positioning of the amino group and the C beta. Okay? Which means the C alpha is fixed right at that place. Anything other than a D amino acid cannot bind is what we proposed at that time. That was around 2013. Then little later, we looked at the mechanism of the system. And what we figured out is that it's not just specific for D alone. It can work on gly also, because we couldn't explain mechanistically how this protein would exclude gly. Gly can very well fit. So we proposed the fact that this enzyme can deplete gly tRNA gly in an in vitro biochemical reaction. We suggested that DTD works on what is called an L-chiral rejection mechanism, or in other words, anything but L. Okay, so it cannot touch L-amino acid, but it can work on anything else, including glycine. And now I am shifting a little bit the gears. So what we talked about is chiral proofreading and chiral mechanism. I am now getting into how the enzymology is done. So one is capturing the substrate. The other one is to cleave the substrate, which means if I have a wrong substrate, like say a D-amino acid tRNA, the D-amino acid has to be removed from the tRNA. And how is it done? So, as I said, the RKL enzyme that we originally worked on and the DTD had similar fold and similar kind of recognition mechanisms. So we thought of now solving structures and doing biochemistry to figure out how the catalysis happens. And then a lot of surprises were there waiting. And one of them was to provide an alternative model to the double sieve. As I said earlier, the double sieve model by Allen First, when he proposed, he said the cognate substrate would be rejected from binding to the pocket. But what we found was something very interesting. That's what I will show in cartoon, because this is a lot of work with mechanistic details. And most of the work was done at that time by Tanweer, who is in the audience and one of the organizers, when he was a PhD student in my lab, and followed by several others. So this is the active site. It's a simple cartoon, not very accurate, but it just tells you what happens. So if you take the active site pocket, you have these side chains. So we mutated these side chains to something similar and showed that the chemistry of the side chains is not important, which means I can mutate the residues to something else, which displaces the chemistry, which means the active site chemistry of residues is not important for catalysis, which means there is something more to it than the amino acid side chains in the active site. And here is, for example, for people who are not structurally oriented, this is adenine, ribose, the rest of the tRNA. This is the amino acid entity, and this is serine here. So here we are talking about an editing domain which is supposed to bind to serine, and therefore threonine cannot bind according to the double sieve model, and serine can be hydrolyzed because serine attached to tRNA threonine is deleterious for translation. So now we will see there's a space, and in that space, what we call as a catalytic chamber, whatever reason, that can hold a water molecule. And that water molecule would be activated, basically, I think it went a little faster. The 2' OH of the ribose is the key here, because of course 2' OH of the ribose is key for many, many important processes which the ribose does, which all of you must be knowing. And the 2' OH activates the water molecule, and that in turn goes and cleaves this ester bond. And that's how the cleavage happens. So this is normal, it's an RNA-based catalysis. Now, I'm going to bring threonine. So in principle, if I make a complex with threonine, it should not work because according to Allen First's model, it should not bind. But what we showed was that, as you hope you noted, the threonine comes in, there is a slight reorientation of the whole platform. And therefore, the catalytic chamber has shrunk so much. There's still some space, but not enough space. And the water molecule cannot go in, and therefore it can be rejected. And just to add a little bit of history there, interestingly, now that in retrospect, the paper was not reviewed by some, so-called high-impact journal, which was rejected, but it went to PNAS, and it so happened that Allen First himself reviewed this paper. He edited this paper. And he said, this is really nice, and that was to be expected. Because you think of now two substrates. When he proposed the model, he proposed it based on basically isoleucine and valine as two amino acid entities. But the substrates of the editing enzymes are not isoleucine and valine. It's a whole tRNA with all the paraphernalia and adenine, ribose, and then we are talking about a simple chemistry of one amino acid, so that rejection will not work as effectively as originally he envisaged. And therefore, such variations are expected to happen, is what he wrote. And therefore, the cognate would be rejected. And that's not the end of the story. When Tanweer left, that was on only one enzyme where we solved the structure. The fun began later. I'm not going to take you through this gruesome structural analysis. So we went and solved structures of multiple enzymes, because this is a variation to the textbook model. And therefore, we wanted to make sure that it happens in every system. It's not just because of one funny corner, one archaea and one organism gave you the mechanism of that type. So we took multiple archaea depending on, belonging to different clades. And the story became a bit interesting, but the message is the same. None of them can hydrolyze threonine attached to tRNA threonine. But the way they operate, for example, in one case, the catalytic water is absent. I will show you a cartoon if you don't understand much here. In another one, this whole substrate is not kept very stable way, so that water can move around a little bit more. In another one, the water was found, but the substrate was kept in a totally different way. So you have different modes by which the rejection can happen. At the end of the day, all of them can bind the cognate substrate, which is different than what Alan first proposed. And okay, so this is just have fun. The sterile tRNA threonine will be accurately positioned. So the non-cognate substrate to be edited from the synthetase point of view is positioned correctly with the water molecule, and then the 2' OH can activate this and this hydrolysis can happen. Whereas in one RKL system, we have a scenario, this is based on multiple high resolution crystal structures around 1.5 Armstrongs and so on, and with different copies, and therefore we know what we are talking about. So here you cannot have a water molecule, therefore the question of hydrolysis doesn't occur. In another case, water is partially present, but the substrate has phenomenal mobility, therefore the catalysis cannot happen. As all of you may know, for catalysis to happen, the distances that we are talking about in terms of positioning the functional groups have to be in picometer scale. If there is a deviation from the picometer scale by 0.1 or 0.2, you can have orders of dip in terms of the enzymatic efficiency. And that is that case. And then here, even though you have a water molecule, because of the variations in the platform, the substrate is kept, the amino acid is kept so out, now the water cannot reach and hydrolyze. So these are the variations that you see, but the bottom line here is cognate substrate can bind, but it cannot be hydrolyzed using other mechanisms. Then came the fun. So we mutated now all the amino acids residues in the binding platform, and we found that none of them killed except for the size-based scenarios. For example, if I have a lysine, if I mutate to an alanine, it will not work because you are disturbing the platform. But if I mutate a lysine to methionine, the chemistry of the lysine is not invoked, but the methionine will have more or less a similar size complementarity so that it can hold the active site. So no amino acid had a direct role in the chemistry. The only functional group present there is nothing but the 2' OH of the ribose. So the moment I make a deoxy, the enzyme became totally useless, or basically it was not functional. And of course, if you do deoxy, people can ask the question that you have a size problem. Again, it can come with certain disturbances because you are, taking out an oxygen group and putting a hydrogen that can leave certain gaps in the area, and therefore that can be a problem. And therefore, we also did a fluorinated substrate, and which cannot be used for catalysis, and which very clearly said that the enzyme is non-functional. So the role of side chains was ruled out. The same we did for the DTD system, because both the systems derive from the same ancestor. One is used as DTD and another one is a RKL editing domain, but they had a common origin, and therefore their modality of function in terms of catalysis is very similar is what we proposed. So when we did the same thing for DTD, the side chains were basically you can mutate to anything and they were functional, and we did the same experiment as that of changing the 2' OH of the ribose to deoxy followed by fluorinated substrate, and all of them actually lost the activity, suggesting that there is none other than the 2' OH of the ribose which is responsible for the chemistry that is happening in the active site. Okay, so that brings us to a bit of a primordial enzymatic scenario, as partly you heard from Jack in the previous talk. If you think of life originated from RNA world, where RNA did most of the catalysis, the early proteins could have been just peptides working together with RNA for a purpose. So what we proposed was this mode of operation that we see in the case of the DTD fold in two different scenarios is some kind of a vestige where the peptide can have significant sequence heterogeneity, but still by having its association with the RNA can perform catalytic function. So that is something which we proposed as how early catalysis could have arrived, and this is actually seen by many. So there are comments by Scott Strobel. If you want, you can go back and read these papers. They're fantastic comment. These are not papers, but commentary on a large body of work, how metal ions are involved in catalysis. And then another one by Sidney Altman. Over here, what he says is, we generally, in the late 80s and the 90s, the thought process was that we are in a protein world. We moved from an RNA world to the protein world. But what he says and what is true is that many of the fundamental processes that you look at, like ribosome, spliceosome, and many others, they still use RNA for catalysis. So the RNA is the crux of how the functions are happening. And therefore, we have not moved completely to a protein world. Of course, it's true that protein have taken over majority of the functions, but still the core functions are retained with RNA, which we don't know how many million years or billion years. If we survive that, they can come to, they can be obsolete. But at this moment, what he suggested, which is very true even today, is that our current world is a protein RNA world and not a protein world alone, is the suggestion. And a DTD-like fold, basically whatever I told so far, is like an intermediate in protein-based enzymology evolution. And you may want to think of it this way. I would also show it in the final review. This is a very comprehensive review looking at all the RNA-based catalysis and how the protein component plays a significant role in terms of aiding the catalysis. So if you look at the ribosome, of course, you can think of ribosome as a large RNA factory wherein the RNA has to be stabilized, because RNA structurally can actually have fold, but it may not be as stable. So the protein components were more invoked for actually stabilizing these RNA components so that you can be much more stable to function in a cellular environment. Whereas the DTD fold where I showed is actually a collaboration between a peptide or a protein and an active site of an RNA, where the protein plays almost no role other than providing a platform for substrate binding, where the RNA now takes over for the catalysis. So it's a nice collaboration in terms of RNA-protein hybrid for a catalysis. So in essence, what I wanted to conclude before we go to the next talk with a small break, I guess, is that chiral proofreading can be invoked alongside these with an idea that it is very essential to bring in a proofreading for D amino acids from a very early times, which means we are talking about from a chemical evolution to early biological evolution scenario. Once that perpetuation happens, the chiral proofreading has to happen throughout. And therefore, in translation proofreading today, you have all these kinetic proofreading, of course, is more of a process in both cases. And the side chain based or chemical based proofreading, which was originally invoked by Paul Berg, Alan First, and Paul Schimmel, and so on and so forth, they become very important for looking at very similar substrates. And of course, when all of these go to the ribosome, you have a whole story of ribosomal proofreading that happens later, which I'm not going to touch with other people. I don't know whether they're speaking or not. The ribosomal proofreading is covered here in some form.
Yes,
so that's again, it's another very important area of
5-minute break.
I'm going to stop here. I'm just looking for the stop. Okay. So the summary so far here, what I said is that we covered early and current mechanistic view of proofreading. They introduce you the concept of chiral proofreading and how it operates. Basically, how the RNA is playing a significant role in the modules, wherein the protein plays in terms of providing only the platform and suggesting how early catalysis in a protein RNA hybrid could have possibly evolved, which is taken over by proteins, as we see, for more effective catalysis. So with that, I stop here and we'll continue the evolutionary implications of chiral proofreading a little later.
Break, we can ask some questions.
So are we having a proper break or we don't have break?
Inside the room.
Okay, fine, no problem. Then let me sit.
We also have a question.
Oh, yeah. So there are many racemases which actually tilt the pool of L amino acids to P, which is one. And there are also things which are amino acid oxidases, which actually can convert from D to L. And many of them are not very specific because each of them can do for 3-4 D amino acids. Like say, they are specific for hydrophobic residues in one case, and there are hydrophilic residues, there is one enzyme, but you do have these enzymes across the board. And just to conclude on that question, that you see, even the L amino acid concentrations, if they go a little imbalance, that also can create problems in the cell. So what the cell does is convert the L to D using these racemases and keep them as reservoirs. So that also happens in the process. Yeah, please, share. They don't, but you will see all of them, correct? They are not essential to look at, but they are under stress. So, essentiality itself is now questioned, correct? I mean, it may not be essential under one growth condition, but it's conditional. Under a condition.
A lot of effort goes when output transcription is regulated.
Mm-hmm.
So in those cases, to crosstalk with other pathways so that DPP can be upregulated in disease of stress.
No, we have not looked at DTD's interaction with others under stress condition, I should admit. I also do not know, because only very few groups working on that. I don't see anybody who has done that. But one can possibly expect such things to happen because our orientations are a little different. We didn't go that way. Particularly now, we have these mice which we have generated over a period of time. And we are, of course, looking at from the cell line point of view. There, the D amino acids are in real excess, and these are neuronal cells. And therefore, certain behavioral things can be looked at. And we are planning for a program with someone where we can look at the interactions. Jisha, I hope you are listening to me, what I'm saying. So, are we doing that? Yes, okay. Note it down otherwise.
Hello.
Just
Quick
yeah, please,
question.
yeah.
Hello. So you mentioned bacteria where two synthetases were lost. I mean, not lost, they don't exist, right? So other enzymes have to step in, which seems to be unnecessarily complicated. So is it known if these enzymes were lost during evolution or they were, they haven't evolved in this specific clade? Because it's kind of very puzzling for me, an interesting aspect.
So there are two ways of looking at evolution, correct? One is that you can think of them are lost in certain branches. That's one way of looking at from a LUCA, because LUCA you would expect all of them to be there. So here it is majorly gram-positive. So what people propose, this is the work of Dieter Saal and co-workers, as you know. What they propose is maybe asparagine and glutamine were added little later to the genetic code. Correct? They were not the early amino acids, and therefore there's a possibility that they are not there in this organism. But if you ask me, frankly, because all these are gram-positive organisms which have come after a LUCA, unless otherwise they have come from a separate branch, which I don't think there are enough evidences. So in that sense, I would say in these branches it were more lost than they were, I would propose the other scenario.
But they can accept
Thank you.
extras. I mean, if you bring in GlenRS from outside, they will accept it. They'll work.
Good for them.
Yeah,
Thank you.
That they work, yeah. The synthetases are non-discriminatory there, that's how they survive. There's a question here.
Yeah, hello.
Yeah.
Okay. Okay.
Mic is working. Just click, tap.
Yeah, so these are huge proteins, the synthetases.
Yes.
So apart from their function in, say, charging the tRNA, do you think there is some other function also?
Oh, yeah.
And
So,
if the mutations in the activity which affects the charging is independent of other functions?
yeah, so there's a whole, battery of people today who have shifted from the core synthetase functions to non-canonical functions of synthetases. And they want to add
20 minutes.
Okay.
You can start just.
Do I get a breather?
Hello.
You have a whole battery.
Yeah.
Yeah,
So
please go ahead. Yeah.
So the question just want to add is that you told that there is both like L and so you have told about only DTD, but then there are several proteins you also told for all domain of the life there which have D amino acids, and then you also told that you can't have a protein with L and D combined. So is there any LTD also like which?
So that's an interesting question. So if you were to think of an organism which were to only put D amino acids, Deepa would say that we should not do.
No, I am not saying about the organism.
We don't have LTD.
Okay,
There's
so
nothing like it.
then
Because
how-
LTD, imagine now if you have an LTD in a biological system, it will deplete now every L amino acid which is attached to tRNA. That means you are not going to have life, which means you can't do any protein synthesis. Is that clear? Maybe I think we should continue because Umesh has
Yeah.
given me a deadline of when to finish. Okay, so I hope, but maybe give me a few more minutes, Umesh, if possible. So I'm going to start off the second part of my talk with the chiral proofreading and its evolutionary implications. So less of mechanism, more of evolution now. And we'll start again from Paul Berg. It looks like Paul Berg has touched everything in this area in a very significant way. And then with another person who is another guru of modern molecular biology, biophysics, which is Donald Crothers. And we'll think about how this achiral molecule, which is glycine, that connects the work of these two giants, which were done independently almost 50 years or so back. They had no connection, of course, other than personal connection. They had no scientific connection. These two works were independently done, published independently, and they were remaining there. And some of the data were sitting as outliers in their data, in their paper. But how our work after 50 years connected those two outliers using an achiral molecule, which is nothing but glycine in the context of protein synthesis. And how that connection has a lot of implications for evolution of the life as we see today. So that's what we are going to see. Okay, so this is Paul Berg, as many of you know. So he identified first a D-amino acid removing activity from tRNA long back. And in fact, Paul himself told us in one of the meetings through Paul Schimmel, I didn't meet him, but through Paul Schimmel, that these were times, where people were not sure L and D are kind of contaminated, during your preparations. So, and then this whole molecular biology bandwagon was gaga on what you call L-amino acid protein making. So what is the use of D-amino acids? It's very likely to be a contaminant. So that work was just left as such. Nobody looked at this DTD protein for a very long time, almost for 35, 40 years. So in his work, while he said that this protein, which he isolated, bucket biochemistry by Callender little later wrote a commentary on our work. Berg was supposed to sign, but he didn't sign because he didn't do anything to that paper. These are high ethics guys. So Callender and Berg were supposed to write a commentary on our work, but only Callender wrote something. Berg didn't write at all. So he said that now that I didn't even contribute because he was indisposed, that I can't sign. Otherwise, I would have signed, what he said. And he reported this outlier activity, which the DTD can actually have some kind of an activity on glycyl tRNA. He didn't talk about it in that paper. The focus was on D-amino acid removal. So he showed that there is an activity in the cell which is done by this enzyme on removing D-amino acid from tRNA. That was it. Here comes Crothers. Crothers was trying to give some kind of an explanation for why we have these 20 amino acids and why we have these 20 tRNAs. He was scratching his head just to give an explanation along with Sol and Seno. And his idea was very simple, you will appreciate. So what he did was, tRNA has 76 nucleotides, all of you know. And he looked at, after the CCA, right, the 5' you have a CCA, before that there is an unpaired base, which is called the discriminator base, which is shown here. Correct? 73rd base is the discriminator base. So he put together the discriminatory base of all the amino acids. And he found something very interesting. If you have an adenine at the discriminator base level, and mind you, this was done when there were only very few sequences. There were totally 40 to 45 total tRNAs that were chemically sequenced. That was what was available, mostly from bacteria and some from yeast. So using that information, when he aligned, he found that if I have an adenine in the discriminator base, they are mostly hydrophobic amino acids. If I have a guanine in this place, you have mostly hydrophilic amino acid. So the early RNA-based peptide making, protein making systems would not have cared much about what I am putting, because I want a hydrophobic property or a hydrophilic property so that you can make some functional folds. And then of course specificity can be improved upon. Therefore, if I have such discriminators, I can actually separate in terms of the amino acid function, is what he proposed. However, there are exceptions that he didn't talk about because that was not the concern of that paper, but the exceptions were reported. Here also the exception was reported because nothing was talked about. Here also these exceptions were reported. We now know the reason why His and Pro have U or C, whatever, but I'm not going into. The interest here is again glycine for us. Because glycine has a U and this is a bacterial tRNA, mind you. But he didn't know whether it was bacteria or eukaryote, but he just proposed that. And as I told you during the mechanistic explanation, that DTD can actually bind to glycine. And I hope I have the biochemistry here. Yes. Okay. So if you look at the biochemistry of the enzyme, imagine if the DTD were to remove glycine attached to tRNA glycine as efficiently as any D amino acid, then you don't have glycine for protein synthesis. Correct? You will deplete glycine from tRNA glycine as effectively as a D amino acid. So what happened was, in the sequence, if you look at, the discriminator base is U. If I now shift the discriminator base to A, then the DTD becomes hyperactive on the A containing tRNA. But U containing tRNA had 100-fold less activity. And this activity difference is what was partly noted without any explanation by Paulberg also, that it can work on glycine with the bacterial tRNA having U, but the U reduces the activity by 100-fold. So that is some kind of a discriminator code that possibly the DTD has rooted in bacteria to begin with. And that's what I hope this is clear. Is it clear? So the one base shift at the discriminator level can change the activity by 100-fold. And so we aligned now all the tRNAs because now we have multiple tRNA sequence, unlike the case of Donald Crowther's where they had only 40 of them. When we aligned, I'm putting tRNA specifically here, but many other tRNAs have mostly purines. So whatever these guys looked at from 40 tRNAs was a good representative of what happens with the rest of the genome. Mostly purine. I'm putting tRNA which has a for a particular reason. And what we showed was that this U here is responsible for glytRNA fidelity for protein synthesis, because if I now do something else by changing the tRNA, then I can actually, you'll show you data that can be harmful to the system. So the U as a pyrimidine discriminator base is key for bacterial protein synthesis. Now, if that were to be the case, is DTD's action on glycine, on tRNA glycine, or glycine on any tRNA, is it a mechanistic flaw, or it was frozen during evolution as an accident, or it has any functional role? This took us several years to figure out. What we showed very surprisingly was that tRNA alanine can be charged with glycine by LRS, because LRS does two mistakes. One is serine, another one is glycine. Paul Schimmel's group had shown long back that the serine removal is so important for the cell because glycine removal did not show as much of toxicity as that of the serine removal. And therefore, glycine may not be important is where they finished based on their in vivo data. What we could show was DTD is a much more stronger deacylator of glycine attached to tRNA because, as I showed you, the discriminator base there is an adenine. And therefore, it can work as effectively as on the D amino acids. And therefore, there is an additional role for the glycine activity of DTD, and which is functional, and we have shown it in systems. And that was what we showed. So basically, removal of glytRNA activity with A as the discriminator kind of connected the two observations made, but which were not explained at that time independently. So this work kind of connected. Now let's see. So what are the implications of Berg-Crowther's achiral connection for evolution of life forms is what we are going to look at, even though we didn't look at it from that angle. I'm just projecting it in that manner. Now we come to, after 2 billion years, the eukaryogenesis, which is beginning. So we now know that which Asgardorgia consumed which bacteria, which is alpha proteobacterium, and that too closer to a rickettsial based on all the genome sequencing and comparison to eventually give rise to the eukaryote that we see, which happened around 1.7 billion years ago. Now the split here becomes very interesting. You see the DTD1 is present in all of eukaryotes. The archaeal DTD2, archaea has a totally different system. So it looks like every biological system requires this activity, but you have two independent evolutionary events. So one has a DTD1, another one has a DTD2. Why, if this activity were to be so important that nature has found two independent solutions rather than one solution at the level of last universal common ancestor, is still a larger question. We do have some answers. We think we have figured out one common DTD at the LUCA level, but that's work in progress. But for time being, that you take it for granted that you have a DTD which is independent of DTD1 in terms of sequence and fold, but does the same function. And is distributed in this manner, where plants are the only organisms which have two DTDs. Okay. Now we go further. Okay. So here comes, I have to rush a little bit. So now, when it comes to eukaryotes, so the first part of this, when I'm referring to eukaryote, I'm talking about the Ophistochondra branch of life. So Ophistochondra means here all fungi and all animals, no plants at all. So this eukaryote refers to Ophistochondra, so all fungi and all animals. So they have only one DTD, but you get two tRNAs because the cytoplasmic tRNA, which is nuclear encoded, is derived from the archaea. This sequence-wise is well known, correct? Whereas the mitochondria, the sequence, tRNA glycan sequence is coming from here. It does not have U, but I'll tell you. The crux is that how, when you are going to have eukaryogenesis, eventually you have only one DTD in the system. How is it going to operate with two different tRNAs, which are going to have different discriminator codes, because one of them will be at cross purposes. Okay, so the bacterial DTD is now the one which is nuclear encoded and goes to both bacteria, goes to both the cytosol and the mitochondria. Okay, so first what we did was to express the DTD protein from bacteria to eukaryotes, just to see the compatibility. And you can see it's very toxic to the yeast. Whereas a catalytic move toxic. And what can be also seen here, of course, using the famous urea gel assay, which Umesh taught us, we could show that the cell death is nothing because of glide tRNA glide depletion, which means there is no glycine tRNA available for protein translation. And we checked other tRNAs and they were all retained. So suggesting that the glide tRNA depletion is the key, which means DTD1 from bacteria is not compatible with the eukaryotic system. And we went to the eukaryotic system and looked at the eukaryotic DTD's activity. I'm showing always one, but you take it from me that we have done it on multiple systems. The eukaryotic DTD has just reversed the activity towards the discriminator, whereas the bacterial one has a high activity towards adenine and lower activity, which is 100-fold difference in terms of the numbers. Whereas that has shifted, which means this has high activity towards U73, which is pyrimidine, and low activity towards purine. And that shift is essential. And therefore, when we now take eukaryotic DTD, I'm putting it in the mitochondria, but in principle, it is the bacteria, correct? If I take it to the bacteria, I can show that the eukaryotic DTD also is not compatible with bacteria. And therefore, the discriminator base has shifted, and therefore it would deplete gly on tRNA gly, and that's exactly what has happened. And now this is a table which you will see in a more complicated ways as we go along. This is, I'm putting now all these organisms of Ophistochondra branch along with bacteria, and we are looking at the discriminator code of all the amino acids. Here you see mostly purines here, except few. And the glycyl tRNA is the only one that has shifted its discriminator code when it moved from a proper bacteria to a mitochondria of a eukaryote. No other discriminator code has shifted from a pyrimidine to purine, because pyrimidine is generally preferred for the discriminator base, And there are several explanations. One of the very common explanation is if you think of a CCA adding enzyme, if you have a C or U in that position, the CCA adding enzyme makes more mistakes. So it puts multiple CCAs. So therefore, and then the U pyrimidines don't stack well with the topmost base, so that can create instability in the stacking. And therefore, that's the reason why purine is always preferred as the discriminator base, because there's a free hanging base at the top of the base pair. So this is the only thing that shifted. Now, so what we could very clearly show using these experiments and then different variations to it, that when the DTD1 moved from bacteria to the eukaryote, the discriminator code shifted from a A-specific one to a U-specific one. Therefore, it's compatible with the A-containing archaeal tRNAs. And the U73 of the discriminator of mitochondrial tRNA gly, or bacterial tRNA gly, shifted to UA, and that's the only tRNA that shifted its discriminator code when it became a mitochondria. And this shift in the discriminator code is essential for mitochondrial evolution and eventually the eukaryotic evolution. Of course, there are many processes that would have been kind of optimized when you moved from bacteria or single-celled organisms to eukaryotic organisms, because imagine these are two systems, archaea and bacteria, that have evolved independently over 2 billion years, and they were brought together to make a new organism. So which means you expect to have conflicts half of the life. Okay, so so that is with respect to this, which of course interestingly, I didn't touch about that, we have no time. There are organisms called Jacobites, and Jacobites are, for people who are working on fungi should know this, they are the very early diverging eukaryotic branch around before yeast, before fungi. And they have the largest mitochondrial genome that we can see for any organism, because some of them have close to 110 genes coded, and their DNA, which is coded there, is much bigger. Right? The genome is much bigger. And people use Jacobite as a model system for very early bacterial, ancestor that has been consumed by an archaea, and that becomes a template for us to look at how the mitochondrial evolution happened from a slightly larger mitochondria, because you know that bacteria codes for close to 4,000 or so genes, and that has been reduced to something like 40, 50 as we see in yeast and higher systems. But many of these genes have been transferred to the nucleus. The mitochondria can never get a gene back, but therefore close to 1,000 to 1,500 proteins visit the mitochondria, which are transferred from the original bacteria. And that's how the mitochondrial proteome is much bigger than what it encodes for. And so Jacobites present you a very unique scenario. So when we looked at the tRNA sequences of Jacobite, what we saw very interestingly, in bacteria you have two of these tRNAs, one U containing, one A containing, or both U containing, sorry. When it moved up to Jacobite, one of them converted it to A, and the U containing one was lost as it moved from the Jacobite to the yeast branch, or to the other Opisthokonta branch. So that means we could even see when this evolutionary transition happened, and that is at the level of a Jacobite. Okay, so now we come to the plant systems, which again makes the case very, very interesting, because as I told you, they have two DTDs, and plants also are interesting because they have a second primary endosymbiotic event that happened after the mitochondria, where the cyanobacteria became the chloroplast. So now you are going to have a conflicting situation that once you have optimized, you are again bringing one more bacteria inside to make another organelle. How is this trade-off going to happen? So bear with me for this. It's going to be a little complicated, but I'll take you through. You don't have to worry about all the rest. Just look at the glycine discriminator. This is plant organelle, Ophisthokonta, which means fungi, and animal organelle. This is archaea, and this is eukaryote. While the Ophisthokonta organelles shifted the discriminator preference to A to compensate for the DTD1's shift in the discriminator code, the plant organelles did not switch. Right? So that was a mystery. And this is based on tRNA predictions from thousands and thousands of genomes. We are not talking about sporadic tRNA sequences. And therefore, this did not change. So how the plants solve this discriminator code issue was the next major question that we asked. Okay, that's what I asked. Okay, so the first and foremost, we took some help from a plant group which was next door. We generated DTD1, DTD2, which is also present in plants, another complemented lines. And we looked at the DTD1's localization because the DTD1 has shifted its preference, so it should not be compatible with the organellar tRNA glycine carrying U in the discriminatory position. So when we looked at the localization, we saw that DTD1 has absolutely no localization both in the mitochondria as well as in the chloroplast. These are all published work, so we can refer. I'm not going to go into the details, but the bottom line is DTD1 is not localized in the organelles of plants, unlike where the DTD1 can go into the organelles of the Ophistochondra branch. Okay, so the next is, okay, what happens if I now push DTD1? So we made these vectors so that with the different targeted sequences from 2 different plants, we now injected the DTD1 to go into the plants. And of course, I'm not showing data here. We did the mutants of DTD which are non-functional, which are not lethal, but however, you can see if the proper DTD of the plant one, which is restricted only to the cytosol, if I now push it using an organellar targeting sequence in the plant, you get severe toxicity, suggesting that you have serious tRNA depletion. And then if DTD1 doesn't go, who solves the problem of organellar chiral proofreading? Because organelles have been shown to possess D amino acids recently, and you will see all the D amino acid present papers were all over the last 15-20 years because chiral chromatography has improved its sensitivity. And therefore, that took us to see whether DTD2 in principle can do this job. And just a small introduction of DTD2, which we worked for some other reason, but it came back here for organellar localization as well. So we were looking at why plants should have 2 DTDs, and this is just a summary of the whole work. I hope I'll be able to convince you. See, plants are the ones which face significant amount of D amino acids compared to any other species because of the microbial degradation that happens in the soil. And therefore, the roots are always in the presence of significant amount of D amino acids. On top of it, the plants also go through what is called anaerobic fermentation that produces significant amount of acetaldehyde. Okay, because they go through ethanol pathway, and therefore, eventually this acetaldehyde accumulates in plants. And the 2 of these, both D amino acids and acetaldehyde, the combination of it, what it does is that the D amino acids which are attached on the tRNA, which are not recycled yet, because acetaldehyde can diffuse much faster than an enzyme, they can convert the D amino acid on top of tRNA to an ethyl adduct. The amino group of the amino acid of the tRNA will be converted by the acetaldehyde to an ethyl adduct. And that is a dead-end product for sure, and there is no enzyme biochemistry known which can remove those adduct amino acid from the tRNA. Okay, if you don't remove, you have adduct accumulation, which you have shown in multiple plants, at least 2-3 plants. And also, the adducts are also dangerous because the plant cannot grow, the plant dies. So what we showed was the DTD2 protein was acquired from which methylogenic archaea also we know because these sequences are available. So this DTD2 is horizontally transferred to what are called charophytes, and these are called land plant ancestors, particularly to a system called chlorokybus, which is a subaerial land plant ancestor, which went on to become a land plant. So this recruitment of DTD2 protein to a subaerial chlorokybus, which is a charophyte, as a land plant ancestor, is essential for the development of land plants, and that's how land plants started migrating from water to land, which was followed by animals. So this is the process called terrestrialization of land. Plants moved around 1 billion years ago, followed by the animals later after the plants went out. So this is a key event in the emergence of land plants, and that's what we showed in the case of DTD2, which is present uniquely in plants, which was coming from archaea. Now, so obviously, what is DTD2 doing? If it were to go to the mitochondria or chloroplast, whether it works on glycine and how it operates with the discriminatory code. So when we did this, unlike DTD1, which is a L rejection system, It works on anything but L, that's why it operates on gly. DTD2 does not work on glycine. Okay, it works as a dechiral rejection system for its enzymology, and therefore it works with gly, but the difference in affinity is 4 orders of magnitude. In a cellular scenario, it makes no difference. So DTD is a dechiral rejection system, and therefore it will not work on glycine even at very high concentration. So in principle, DTD2 can go into the organelle, and is that the situation? Okay. And that's what this data shows. We generated a DTD2 complemented line in Arabidopsis and looked for DTD2's presence, and you can see in addition to being in the cytosol a little bit, it can go into both the organelles. And you can see a very high, what do you call, co-expression or no, both of them can be present simultaneously in the system. So basically, co-localization, yeah, that's the term I was looking for. Co-localization in the organelles, okay? So, which clearly says that DTD2 can go in while DTD1 cannot go in. That kind of resolves the discriminator conflict in a different way. Now, okay, so this is just a summary in case if you have missed out somewhere. I hope I will be able to take you through. So here we are talking about a bacterial system where the DTD1 of the bacteria can work on D amino acids, and it also works with glycine. And it is compatible with the U discriminating base because DTD1 cannot deplete glycine on tRNA glycine because the U is antagonizing DTD. Fine? Whereas the DTD2, it doesn't matter what the discriminator base is because it doesn't care about the discriminator base, and on top of it, it doesn't work on glycine. So it need not worry about tRNA glycine. Now, when you go towards eukaryogenesis, what goes to the archaeal, the cytosol of eukaryotes is the archaeal tRNA gly, and that goes with A as the discriminator base. Now, the bacterial DTD1 shifts, I hope you saw the change in color. Okay, it shifts its discriminator code in such a way that now it is compatible with A because A is antagonizing and U is its preference, and therefore it is compatible. And now the bacterial tRNA gly shifted its discriminator base from U to A, and therefore it's now compatible with the DTD1, which can go into the organelle. That's what we saw with the bacterial work. Whereas the plants, for some reason, which we don't know yet why it has happened this way, has not shifted its discriminator base. And the DTD1 cannot go in because you push VC toxicity because they are incompatible. And therefore, the DTD2 is horizontally gene transferred from archaea, and that is pushed to the organelle, and therefore now this is compatible. So this is what we have shown so far. In the context of Paul Berg and Donald Crowther's findings, there are 2 outliers in the data done independently 50 years back, and very clearly suggesting that if you have outliers in data, you are supposed to put it somewhere so that can come to use. And we have put a review on this, which if you are interested, maybe you should go and look at it, when Paul Berg meets Donald Crowther and how using glycine made this achiral connection during protein synthesis. And as I was mentioning, significant research, of course, can come back in multiple ways, correct? It doesn't matter which journal that you publish, they have a way of coming. Impact factors are only 30-year-old stories. There was no impact factor in biology or anywhere 30 years back. And the importance of outliers, right? Always put outliers, even if you don't explain. That's what these gurus of modern molecular biology told us, that you may not use it, but the outliers, if you can't interpret, somebody can actually figure out the use as long as the outliers are correct outliers by doing the correct set of experiments. And it also shows, the unbiased way of reporting by these guys, correct? They could have just hidden that data because they are not fitting in their larger kind of framework that they are talking about. They could have just left these, but they brought it in and showed that, okay, this is what it is, but they are not even talking about it. That's very important. Of course, your third reviewer will kill you. We'll have to put up with them in today's world, but that's how life goes. Okay, so just to summarize, now we have lots and lots of genome sequences because multiple evolutionary intermediates are sequenced. Of course, the Chinese guys have taken a great lead in this. Every organism is being sequenced and you get to see a lot of cross-genome comparisons. So this is a very fertile ground for people like us who are looking at biological processes, whichever process that you are interested in, how these processes have evolved in different niches. And is there any kind of cross-compatibility which could have possibly set in at some point when you move from one organism to another organism, because you have the genomes of all the intermediates getting available. See, the same work on DTD2, if we had done, I would say even 5 years before, we would not have interpreted the data that you have a horizontal gene transfer that happened from methylogenic archaea to chlorokybus, because none of their sequences were actually available. You understand? So in today's world, we could interpret as to from which organism to which organisms things actually can move. And that can be very valuable for us to understand the evolutionary transitions. And of course, that's what I wanted to say. Okay, so you may ask me, all these are translation, because in today's world, if I go and say translation to somebody, they think of something else, how much fund, right? What are you going to translate? Right? This other translation is going to the back burner, and unfortunately to some of our biologists also. So, but is it all translation of the genetic code, or our work has anything to do with the modern usage of the term? I just thought I'll show you this data. So we are very excited about when you overexpress DTD2, it kind of gives you phenotypes of stress resistance, the plant stress resistance when it happens. These are the kind of root phenotypes that you get, which kind of resistant to multiple stresses. And plants, which we realized over a period of time that are the organisms other than archaea, are flushed with aldehydes. There are different types of aldehydes, and their concentrations are way, way higher than what a bacterial system or a eukaryotic system can handle. And therefore, this aldehyde detoxification becomes very, very important. And we have some efforts on this to see if we can take these proteins towards improved crop development and so on and so forth. So to conclude my presentation, what I have shown you, that these chiral proofreading molecules are involved in many, many different critical phases of evolution. One aspect which I didn't show you, you may have noticed in the distribution of these chiral proofreading systems, something called ATD, which is called Animalia specific tRNA deacylase. That's a name that we gave to it because it was present only in Animalia. Just hint what is the problem. If you go from unicellular eukaryote to higher eukaryotes, the number of tRNAs expanded from few tens to few hundreds, and in certain cases to few thousands. tRNA is a very small molecule, 75, 76 nucleotides. So how many variations that you can generate? So when nature generated these, what we call now the general tRNAs as isoacceptors, there are these new tRNAs that have come, which were called as isodecoders, and which means they carry the same anticodon, but the body can change. Because the body can change, it can now create cross reaction with some other synthetase or some other biological entity. And that's what has happened with one of the tRNAs, wherein this isodecoder generation has created a problem of mischarging. So that is the first problem of not amino acid mischarging. The synthetase picks the correct amino acid, but takes the wrong tRNA and puts it. So ATD, what we call as Animalia specific tRNA deacylase, is a proofreading factor to correct the tRNA-based error correction mechanism. And we showed that how this protein could have, one of the proteins could have played a significant role towards multicellularity, because that tRNA expansion was associated with multicellularity. And okay, and the work, so on and so forth. And it strengthens the ancient view, the view that the ancient biomolecules, because these are supposed to be the ones which are there with us from day one, correct? And they can be used for multiple functions. And also, whatever I showed, hopefully convinced how this coevolution or the dynamics, if you want to think of it that way, between the RNA and the protein players can happen in different niches. With that, I would like to thank all my group for their long-standing contributions. I think Tanweer should be somewhere here. Okay, he's one of the past members and very early members of our team who did the archaeal work, and then several others who did. Nisha is attending the meeting, so you will hear from her more. This guy is down with dengue, so he is supposed to be here, but he's not coming. But I think his poster also who will present, I guess. And thank all the funding agencies. And these are some of the references. If you guys want for further reading, you can take it. Thank you all.
We can take one or two questions. Yeah, go ahead. Okay, she has the mic.
Am I audible?
Yeah, please go
nice
ahead.
talk. So I wanted to ask that you saw DTT2 overexpression has a crop enhancement phenotype. Does DTT1 overexpression also have that?
No.
Or you didn't try?
Of all the talk, you went to the translation component of it, which is anyway fine. The DTD2 can be overexpressed because it does not cross-react with any glide tRNA. Okay, because it doesn't work on tRNA glide, because the orders of magnitude are very different, and I don't think we can express a molecule in a biological system to 3 or 4 orders of magnitude more. DTD1 overexpression has a problem which we have seen in multiple systems. So obviously, to answer your question, it is not going to be useful for crop development or any development for that matter, because DTD1 overexpression will kill the system. Because now it will start cross-reacting with the U, because it has only 100-fold difference in affinity. So the more and more I overexpress, it will react with it. And DTD1, the crop development function comes because of its detoxification with acetaldehyde, modified adductive. Yeah, and that activity rests only with DTD2 and not with DTD1.
Okay.
Am I clear? Okay.
I have one more question. So how different is DTT1 of plant from the other DTT1 that can go into the organelles? Like structurally or?
The DTD1, if you see, which is present across the board except in archaea, is sequence-wise and structurally highly similar. Very similar, okay? Except that the discriminator code, when it moved from bacteria to eukaryotes, shifted.
But the discriminator code is on tRNA.
No, the discriminator code recognition elements are in the protein, correct? So the discriminator code, of course, you are absolutely correct that it is with the tRNA, but who is going to recognize? So when I say change in color of DTD1, the one that was possibly preferring A has shifted from U. So there are elements in the protein, which I think my student Tanweer still asks me whenever I see, where are the structures? Hopefully we'll bring one day. The last 10 years, we are running behind this biology.
Why don't you start calling it DTD1? There will be no confusion.
No, it's okay. It's a way of terminology. So the bottom line here is that the DTD1 protein has certain elements that shifted when it moved from bacteria to eukaryote. And those elements, we roughly know where it is, but only thing that can answer is a co-crystal structure of the complex or co-cryo-EM structure of the complex. There's one more hand, one last question.
Well, he will ask you during, or she will ask you during lunch.
Okay, fine, yeah.
We have to break for lunch because we are running late.
Sure.
The lunch will be on the first floor of wherever we had breakfast, or in the canteen first floor, the lunch will be there. And Shankar actually had to go to amino